reducing the plaque depositions in our blood vessels. So it is also benefited in cancer preventions. Just prevent, it's not avoid, we just preventions. So antioxidants neutralize the substances that can damage the genetic material by the oxidations. So here, what I want you to know is the classifications of antioxidant. There is the two types of antioxidants: enzymatic antioxidants and non-enzymatic antioxidants. So if your research final project, S2 project or S3 project get along with the antioxidants, my questions is which antioxidant that should belong, either by enzymatic or non-enzymatic. Okay, normally students always tell me, they will inform me, doctor, I want to run antioxidant. Then I ask, what type antioxidant you want to run? Is it enzymatic or non-enzymatic? So you have to know specifically. Then if you know the specific, you will know what is your pathway to get the antioxidant mechanism. Because different type of antioxidants, for example, enzymes, they have a different mechanisms. So below the enzymatic antioxidants, it will have the primary enzyme and secondary enzyme. Okay. Most of the research, I found that in my faculty at University Malaysia Sabah, they always apply the non-enzymatic antioxidant, which includes the minerals, vitamin, organsulfur, antioxidant cofactor, low molecular weight antioxidant, carotenoid, and polyphenol. So this one is the highest trending, the polyphenol. Okay. So the polyphenol, there is two types. Either you want to see the activity of the flavonoids, or you want to see the activities of the phenolic acids. So it depends what type of project you are. So if you are using antioxidants, if you're planning to run antioxidant in your final year project, S2 project or S3 project, so here you have to know which antioxidant that you use. You have to know which one. Okay, so let's say you choose a flavonoid. You want to see the flavonoid activity. So which flavonoid that you want? So there is a five types of flavonoids that we can found the activity. As we can see here, flavonoids, isoflavonoids. Okay, inilah nama-nama dia sampai flavones. Okay, so if your study is focused on the phenolic acid, so which one? You want to focus hydrocyanic acids or hydrobenzoic acids? So each of these compounds you want to focus, for example, minerals, you want to focus the non-enzymatic antioxidant, focus on the minerals. You have to find out which mineral you want to focus. Zinc, selenium. Okay. So this is the classifications. So this one is the pathway actually for your research. So next is how actually the mechanism occurs. There are five types of mechanisms. So first is the oxidative enzymes, and then we do have the ROS for the exogenous sources, as well as unstable free radicals, and that's the stable free radical. To understand this one, I will share a video for you to get a deep understanding actually how the different antioxidants occurs, the mechanisms. Bukan you bagi I the value of activities. For example, the mean value and then the standard value. I want to see the mechanism because mechanism of the antioxidant will relate to the molecular docking. Okay. Let me share video first. Betul kan? Ada. Zoom. Jadi ada.
In this video, we're going to see what free radicals are and also how antioxidants protect us against them. It's going to be an interesting one. A free radical is a highly reactive and unstable chemical species that contains one or more unpaired electrons in its outermost electron shell. These unpaired electrons make free radicals highly reactive.
Kejap, ah. Kesian korang, tu tengok yang sini. Kejap, ah.
In this video, we're going to see what free radicals are and also how antioxidants protect against them. It's going to be an interesting one. A free radical is a highly reactive and unstable chemical species that contains one or more unpaired electrons in its outermost electron shell. These unpaired electrons make free radicals highly reactive because they seek to pair up with other electrons in nearby molecules. As a result, free radicals can participate in various chemical reactions, both beneficial and harmful, in the body and the environment. Excessive amounts of free radicals can cause damage to cellular structures, including lipids, proteins, and DNA. This process is known as oxidative stress and is associated with various diseases and aging. Some of the most common and well-known free radicals include superoxide radical. This is the primary radical formed when an oxygen molecule gains an extra electron. I'll give you one example of how this free radical can be formed. In the mitochondria during oxidative phosphorylation, a small percentage of electrons leak from the electron transport chain. These leaked electrons combine with molecular oxygen to form the superoxide radical. The superoxide radical is an example of reactive oxygen species, or ROS.
This one is the first
Reactive
mechanism.
oxygen species are chemically reactive molecules containing oxygen. Superoxide is an important free radical in cellular metabolism and acts as a precursor to other more reactive oxygen species. I'll give you some examples. Two superoxide radicals can react with two protons to produce one molecule of hydrogen peroxide and one molecule of oxygen. This reaction is catalyzed by an enzyme called superoxide dismutase. Hydrogen peroxide is another type of reactive oxygen species. Hydrogen peroxide can undergo metal-catalyzed reactions, such as the Fenton reaction, involving transition metals like iron or copper to generate the highly reactive hydroxyl radical. The hydroxyl radical can react with nitric oxide to produce the nitric oxide radical and the hydroperoxyl radical. There are other examples of free radicals which we'll not discuss. Let's recap the most famous free radicals. In the right upper corner, you can see the superoxide radical, hydrogen peroxide, which is not a free radical, but it can generate free radicals, as I have proven to you. Next, we have the hydroxyl radical, the nitric oxide radical, and the hydroperoxyl radical. To counteract the harmful effects of free radicals, the body has built-in defense mechanisms, such as antioxidants, which neutralize or stabilize free radicals by various mechanisms, which we will discuss. The first mechanism is called hydrogen atom donation. Hydroxyl groups are functional groups, commonly found in antioxidants, such as phenolic compounds, for example, resveratrol, quercetin, and tocopherols, for example, vitamin E. Let's take resveratrol as an example. Resveratrol contains multiple hydroxyl groups in its structure. When it encounters a free radical, such as the hydroxyl radical, resveratrol can donate a hydrogen atom from one of its hydroxyl groups. Let's say this hydrogen atom. As a result, a relatively stable radical intermediate, called the resveratrol radical, is formed. Simultaneously, the hydroxyl radical gains the hydrogen atom and transforms into water. Although the resveratrol radical is itself a radical, it is less reactive and damaging than the original superoxide radical. Enzymatic reactions may further process the resveratrol radical to transform it into a harmless product. The second mechanism is called electron donation.
Okay. For the first mechanism is a hydrogen donations. Okay, so the second mechanism will focus on the electron donations. So if you want to produce a natural products from the marine fish, you want to extract anthocyanin from the plants or anything you want to extract from the seaweeds also. So you have to know which mechanism that you apply. If your first mechanism you apply, you may produce the harmless products. If the second one, let's see how.
similar to the first mechanism. It's almost the same thing, and you'll see why. Let's use vitamin C as an example. When vitamin C is dissolved in the body's fluids, it loses a hydrogen ion and it becomes ascorbate. Ascorbate is the ionized form of ascorbic acid, and it carries a negative charge. Ascorbate can donate an electron to a free radical, becoming an ascorbate radical. The new radical is more stable and less reactive than the original free radical. The ascorbate radical can dissociate again in water, releasing a proton, making this new molecule a negatively charged ascorbate radical. This new radical can donate an electron to a free radical again, becoming dehydroascorbic acid, which is not a free radical anymore. The third mechanism is called metal ion chelation. Metal ion chelation is a crucial aspect of the antioxidant activity of certain compounds, including glutathione and vitamin C. Let's take glutathione as an example. You can see its chemical formula right here. Glutathione is a tripeptide molecule composed of 3 amino acids, cysteine, glycine, and glutamic acid. The liver is a crucial organ for glutathione synthesis. One of the essential functions of glutathione is its ability to chelate, or bind to metal ions, particularly transition metals like iron and copper. Glutathione contains a thiol group on the cysteine residue, which is crucial for its chelation activity. Sulfur's electron distribution is 2, 8, 6, indicating that it has 2 electrons in its innermost energy level, 8 electrons in the second energy level, and 6 electrons in the outermost energy level. The valence level of an atom refers to the outermost energy level that contains electrons. These valence electrons are the ones involved in chemical reactions and interactions with other atoms. You can see that sulfur has 2 pairs of electrons on its valence layer. When the thiol group of glutathione interacts with a metal ion, the sulfur atom donates its electron pair to the metal ion's empty orbital. This results in the formation of a coordinate bond between the metal ion and the sulfur atom of the thiol group. A coordinate bond is a type of covalent bond where one atom, the donor, donates a lone pair of electrons to another atom, the acceptor. In the case of glutathione chelation, the sulfur atom donates its electron pair to the metal ion, creating a stable complex. By binding to metal ions, glutathione prevents these metals from participating in the Fenton reaction. As a result, harmful hydroxyl radicals won't be generated. The fourth mechanism is called antioxidant enzyme regulation. Metals like zinc, copper, manganese, and even iron can work indirectly by regulating the activity of certain enzymes that protect us against free radicals, called antioxidant enzymes. For example, there's a crucial antioxidant enzyme called superoxide dismutase. This enzyme plays a significant role in the body's defense against reactive oxygen species, particularly the superoxide radical. Superoxide dismutase catalyzes the following reaction. 2 superoxide radicals react with 2 protons to produce 1 molecule of hydrogen peroxide and 1 molecule of oxygen. Hydrogen peroxide is a reactive oxygen species. Still, it is less reactive than the superoxide radical and can be further broken down into harmless water and oxygen by other antioxidant enzymes, such as catalase and glutathione peroxidase. Here's the structure of superoxide dismutase. Metals like zinc, copper, and manganese are essential cofactors for SOD. They help activate the enzyme and maintain its proper function in converting superoxide radicals to hydrogen peroxide. Additionally, certain antioxidants can stimulate the production of SOD within cells. For example, resveratrol has been shown to upregulate SOD expression, thereby enhancing the cell's ability to neutralize superoxide radicals. Another important antioxidant is selenium. Selenium is a chemical element with the atomic number 34. It is a non-metal with properties that are intermediate between the elements above and below in the periodic table, sulfur and tellurium, and also has similarities to arsenic. Selenium-containing enzymes, like glutathione peroxidase, help break down hydrogen peroxide and lipid peroxides, preventing their conversion into harmful radicals. Glutathione peroxidase, in its composition, contains a selenium atom that is located in the active site of the enzyme. Without selenium, this enzyme can't function and the level of hydrogen peroxide and lipid peroxides inside the cells will rise. Now, let's move on to the chemical reactions catalyzed by glutathione peroxidase. You remember the chemical formula of the reduced glutathione. You can see it right here. Let's create an abbreviation for this tripeptide. Let's call this part of the molecule G, which stands for glutathione. The thiol group will stay the same. The abbreviation for reduced glutathione will be GSH. The first reaction catalyzed by glutathione peroxidase will be the breakdown of hydrogen peroxide. In this reaction, two molecules of reduced glutathione react with one molecule of hydrogen peroxide to produce one molecule of oxidized glutathione and two molecules of water. The oxidized glutathione is basically two molecules of reduced glutathione connected by a disulfide bond. The second reaction catalyzed by glutathione peroxidase will be the breakdown of lipid peroxides. But first, let's talk about lipid peroxidation. Lipid peroxidation is the chain of reactions of oxidative degradation of lipids. It most often affects polyunsaturated fatty acids because they contain multiple double bonds in between which lie methylene bridges that possess especially reactive hydrogen atoms. Lipid peroxidation consists of three major steps: initiation, propagation, and termination. The chemical products of this oxidation are known as lipid peroxides. Initiation is the step in which a fatty acid radical is produced. The most notable initiators in living cells are reactive oxygen species, such as the hydroxyl radical, which can combine with a hydrogen atom from the methylene bridge of the polyunsaturated fatty acid to make water and a fatty acid radical. Then, after a reaction called intramolecular rearrangement, we obtain a slightly different fatty acid radical. Now, let's get into the second step called propagation. The fatty acid radical is not a very stable molecule, so it reacts readily with molecular oxygen, thereby creating a peroxo fatty acid radical. This radical is also an unstable species that reacts with another polyunsaturated free fatty acid, producing a different fatty acid radical and a lipid peroxide. This cycle continues as the new fatty acid radical reacts in the same way. When a radical reacts with a non-radical, it can produce another radical, which is why the process is called a chain reaction mechanism. The radical reaction stops when two radicals react and produce a non-radical species. This step is called termination. This happens only when the concentration of radical species is high enough for there to be a high probability of collision of two radicals. Now, let's see how glutathione peroxidase can protect us from lipid peroxides. In the reaction catalyzed by this Q enzyme, one molecule of lipid peroxide reacts with two molecules of reduced glutathione to produce one molecule of oxidized glutathione, one molecule of lipid alcohol, and one molecule of water. The oxidized glutathione formed during these reactions can be recycled back to its reduced form by an enzyme called glutathione reductase, which uses NADPH as
Recording in progress.
In this reaction, NADPH reduces the oxidized glutathione and turns into an ADP positive. The fifth and last mechanism of how antioxidants protect us against free radicals, which I'll cover in this video, is the activation of sirtuins. Resveratrol will be our example of an antioxidant.
The last mechanism.
Resveratrol is known to activate a class of enzymes called sirtuins, particularly sirtuin 1. By promoting the expression of antioxidant genes and proteins, sirtuin 1 activation helps to counteract oxidative stress and protect cells from harmful reactive oxygen species. Additionally, sirtuin 1 is involved in DNA repair pathways, contributing to the maintenance of genomic stability. Activation of sirtuin 1 can enhance cellular resilience against oxidative damage and support longevity. As a quick recap, the main mechanisms of how antioxidants protect us against free radicals discussed in this video were hydrogen atom donation, electron donation, metal ion chelation, antioxidant enzyme regulation, and the activation of sirtuins.
Okay. All right, so there is a file. Let me share the slide first. Maybe hybrid tapi. So here I show the summary just now. So the video will explain actually each of the molecules, what happened on the first mechanisms, the second mechanisms, the third mechanism until the fifth mechanisms. So in your research, in your final year project, in your S2 or S3 project, anyone here involved with the antioxidants? Anyone here? So now, did you get any idea which antioxidant mechanism you belong in your research? Ada idea? Kan masih pening-pening. So now you know that antioxidant is a general terms. So when you go for your supervisor, inform them you get the antioxidant results, make sure that you can explain what happened actually, what activity inside your results that you get. So which mechanisms you belong, you have to know which one. So there is a 5 mechanism, the major one. So which one is you are? Normally for my part, I will belong onto the mechanism 1 and 2 for my part, for my field of interest. So I came here, I just give an overview. So you have to choose which one is yours. I just give an idea. So antioxidant is a general. So don't give your supervisor is just mean value, standard deviation value. And when your supervisor asking you what happened to the activity, so you have to know to answer based on the mechanisms, the 5 mechanisms, which one is belongs to you. All right? Okay. So after you know that, you know which mechanism that should belong, then you came to the measurement or test or analysis antioxidants activity. Okay, normally ada FREP, DPPH, ABTAs. So, mana you pilih? Suka hati. Normally my student suka hati, yang mana ada? Yang mana ada available, okay, I do FREP. Kalau FREP tiada, ABTS. And then, satu lagi, DPPH. So when I ask, kenapa you pilih DPPH? Why you choose DPPH? Then diam. Then the answer is, sebab chemical itu sahaja available, doktor. Then nak je tarik telinga. So bila thesis, you defense your thesis, you defense your proposal. Kalau itu jawapan yang you bagi, what happened to your marks? What happened to your defense? So you have to know kenapa you pilih DPPH. You have to know kenapa you pilih this technique. So I list out here measurements of the antioxidants activity. Kenapa? Ada? It's okay, it's okay. Ada sedikit lagi. Alright. It's okay. So, first, a radical scavenging method. So if you focus on the ROS, you want to scavenge. Scavenging is the tangkap, radicals, free radicals, you want to tangkap. Okay? So if you want to tangkap, you can use the oxygen radical absorbance capacity assay. If you want to focus for the ROS, there is 4 methods available. The first one, oxygen radical absorbance capacity. The second one will be chemiluminescence assay. The third one, DPPH. The fourth one, Cholox. So kalau you pilih DPPH, you belong to scavenge the ROS. So meaning that you will come to the first mechanism just now, the ROS. Okay, understand? If you focus on the non-radical redox, for example, you use a FREP. So is it applicable? Bolehkah kita guna? Kalau tiada chemical, kita guna DPPH. Kalau ada chemical, kita guna FREP. So boleh? Is it boleh? So betul antioxidant, which one that you focus? What type of antioxidant you want to, mechanism of the antioxidant you want to focus? So if you want to focus the DPPH, meaning that the free radicals, you want to study the free radicals. If you use the FREP, you focus on the non-radical redox potential based methods. So it's a different techniques and different yield that you will have. that you will get a different yield and then different mechanism, all is different. So saya tidak boleh terima, for my student, I tidak boleh terima jawapan antioksidan apa you nak buat. Ada di PPH saja doktor. Frame tiada. Then you focus on which one? You said to me previously you want to focus on the ROS, but suddenly you pergi frame, and your study from the first objective until the last objective is belongs to the ROS. So why suddenly you pergi frame? Kadang-kadang I marah juga. Okay. So the next one, metal selations capacity. This one, when we study about the heavy metals detections, and then we are using the extract from the plant. We extract the phytochemical and we want to detect the heavy metal inside the fish or seafood product. I have to study that one. I have two students that doing that one. So the antioxidants will focus. They're using an antioxidants analysis based on the metal selations capacity. Okay, the last one is TPC. Yang ini paling happening, TPC, total phenolic compound. Okay, when we talk about total phenolic compound, I told you just now this one. If you focus on the phenols, okay, TPC, you will run the TPC. However, my next question is, which phenolic compound that you focus? There is a lot of phenolic compound. There's a lot. So, which activity that you want to focus the antioxidant? Kalau for S1, maybe I boleh terima. S2, at least you know a little bit. For S3, you have to know the specific of flavonoids that you extract. For example, okay, doctor, I want to focus on the flavonoid. My next question is, which flavonoid activity you want to focus? You have to know. Sampai dalam-dalam tu. So, maybe you will tell me, you want to see the differences between these 5 flavonoids. That one is for S3. Okay, so that is a different level, S1, S2, S3. So that's a different level actually how we analyze the words of antioxidants. Bukannya bagi saya the number of the mean and number of the standard deviation. Tiba-tiba bagi saya yang itu, and then I ask, is there any significant difference? I don't know lah, doktor. Maybe doktor yang kena buat sendiri, maybe ya. I kena buat sendiri kot, I kena buat significant. The basic for S1, you have to know the significant difference between the value. Kenapa you buat SD? Kenapa you buat mean? What is the meaning of the standard deviation? Bukan suka-suka sahaja tu ada di situ. Okay, so we can see the error also from there, right? Kalau S3 kena tahu what is the compound phytochemical that you focus. Bukan flavonoid, you kena bagi lagi dalam. Alright? Kalau S2, I boleh terima sampai flavonoid, phenolic acid, itu boleh. Kalau S1, I boleh terima sampai phenol, TPC. Alright? So, seterusnya. This is the Ros scavenging method. The first one, oral accumulations. Okay? Kita ada dua. The blue one is the ROS. The second one is the redox potentials assay. So if you click here, you will go directly to the link of the pembuat method there. Okay, you boleh follow. You tekan link this one, for example here. Keluar ke? Situ keluar di. Ya, so tempat I tak keluar slide saja. So you boleh klik terus Okay, if let's say suddenly you have an idea now. Tiba-tiba ada idea, you focus on the antioxidants, suddenly you think that you focus on the DPPH, you suka DPPH, My student sangat suka di situ, DPPH, PrEP, dua benda ni dia suka. So, kalau you focus on PrEP, you boleh fokus kepada Molina. Molina is the, here, bolehlah. Kenapa tempat saya ni? Okay. All right. So the next one, antioxidant evaluations in food model system. Let's say you ada buat product. You develop a product from the maybe krepek. Okay, maybe you produce a product from the seafood. Okay, become a product, gula-gula ka, snack ka. Okay, so this is the measurements model in food system. First, you can focus on the measurements of antioxidant activity in the oil system. Let's say you want to focus on the krepek. You know krepek, right? Chips, krepek chips, krepek ikan, keropok, keropok ikan. Kalau sana krepek, krepek, keropok ikan. Kalau you want to see you want to produce a keropok ikan. So maybe you want to see the antioxidant activity in the oil system. Nah, kita makan keropok, kalau ada keropok tuh kita boleh find the antioxidant activity in oil systems by using the oxygen consumptions. Okay, by find out what is the oxygen consumptions, and then calculated the change in oxidation substrates. Also, you may measure the oxidation products as well as the system change during the oxidation process. So ada banyak jenis kita boleh. So ada 4 results. Just antioxidant. Okay, trust me, just the results of antioxidants you can publish. Tapi bukan Q1. Just maybe Q3, Q4. Tapi just the results of antioxidant. As long as you do the analysis and then analyze the words of analyzing the result. So the words of antioxidant just now, it will give you 4 results. Oxygen consumptions. Okay, you calculate the changes in oxidation substrates. You can calculate the oxidation products. And there is a 4 mechanisms here that you can report. Right? Normally, I will get for the antioxidants results, normally, I don't know here, normally, as a lecturer, normally my student will give me just one test. Sudah. Oh, kejap. Ayo, kena 2 orang nih, sini, lihat sini. Ini masalah Pak Ede nih. Saya share apa? Okay. All right. So the second one, measurements of the antioxidant activity in the model system, emulsion systems, and then the third one, measurements of the antioxidant activity in the muscles foods model systems. So you can, here, what I want to emphasize actually, you can focus on the antioxidant activity in oil and then in emulsions as well as in the muscles of the food. Let's say you buat ikan kering, maybe ikan kering. Kalau kita panggil ikan kering, okay, ikan asin. So maybe you want to see how the antioxidant activity in the muscle of the fish. That one you can run. So the next one, antioxidants activity assay, measuring the oxidation product. So there are a few, the name of assay here. The first one, you may focus on the peroxides value. The second one, conjugated dienes and TBRS, followed by the andesite value, then total carbonyl, and lastly, the free radical
Recording in progress.
Okay. All right. And then I list out the markers, oxidation marker. For examples, if you focus on the peroxide value, R or S, okay, your marker will be hydroperoxides. And then the reagent you may use is potassium iodide, okay, triphenyl phosphanes, thiocyanate for the detections by using, this is the detections method. Okay, suggestion for you. You can use a titrations. If you focus on the potassium iodide, the best method to detect is using a titration method. Yang tukar-tukar color, you know, right? The titrations method, that could be pink color, okay, colorless to pink, pink to colorless, okay? And then you will see the percentage, you will calculate the percentage of the inhibitions activity. Okay, you will give the percentage. That one I told you just now, it's in the table. My student love to give me the antioxidants in the just in table. I don't know here. Number of how you analyze your antioxidant here. Anyone can share? Janganlah malu-malu. Anyone? How you analyze your antioxidant here? For your research. Tak ada yang salah, don't worry. Table. DPPH. And then the results will come in table. Or ID50. Okay, similar with my student. So after this, after you attend this talk, maybe you can find out what is the mechanism behind the DPPH, and then how actually the OH, or is it electron donations or hydrogen donations mechanism. So after this, you can explain more the mechanism, what happened, what is the activity inside, so that you can relate later on to the molecular docking for the S2 and S3. There is a relationship between the phytochemical. All right, the next one. Measurements of the antioxidants activity in the biological model system. We can use LDL cholesterol oxidation inhibition assay. And then we can use the inhibitions of the TNA oxidations and by Niki. Right? And last one, cellular assay for the evaluations of the antioxidants activity. We can apply the hemolysis inhibition assay as well as the cellular antioxidants assay. All right, it's quite deep. For these measurements as actually it's a biological measurements, antioxidants. So normally this one is for the S3. For my level, for my student level, I will give to the S3 level, this one. All right. So for the evaluations in biological model systems, I mentioned just now there is a few, and then you can see the oxidants, for example, LDL cholesterol, it will focus the oxidant on the pH generated, the peroxyl radicals. For the marker, oxidation marker will focus the conjugated dienes, and the detections will be the spectrophotometer. There is a color changes, and then the wavelength will be different. The wavelength as well as the what we call that absorbance. I forgot already. I can see gini siya peak dia. Peak, good. Okay. Okay, after you know what is the mechanism, after you know the antioxidants, I know you know the antioxidants, but after you get an idea, the mechanisms for your own antioxidants, then you can relate to the molecular docking. This is just an examples, okay? The interactions diagram, this one is the 2D and 3D of the docking score result of the indicate the strong affinity among the complex molecule. As we can see, the first one is the 3D surface. This one is carrageenans against antioxidant target. Ini S2, S3 level lah. S3 wajib. S2 ada yang wajib, ada yang tidak. S2 masih digalakkan. For S3, for those yang buat antioxidant S3, please focus on this molecular docking. You want to see the interactions between, just a suggestions from me. Tapi tidak wajib lah. For me tidak wajib, tapi just a suggestions for me if you want to get an impact for your research. Right? So the first one, molecule docking of the carrageenan against the antioxidants target. We can see a 3D surface mass map the interactions of the sites, the mass map of the 3D complex, and B, 3D complex interactions, and labeled by C, 2D complex interactions by the antioxidant and the carrageenans. So you can see the target. Okay. D will focus on the 3D surface mass map interaction sites, and E is the complex interactions between the carrageenans and antioxidant activity. And the last one, actually, how actually the 2D complex interaction. These are some of the examples. And this one is we focus on the S3. Since my audience from S1, S2, S3, so I just let you know, for S1, you focus on the polyphenol. At least you know how to calculate the ID50, or if you, at least you know what method, if you are using in your final year project, if you plan to do antioxidant for S1, this one I focus on S1. First S1, I focus for S1 students, for those yang hybrid pun, yang ada dalam Zoom, alright? For S1 students, at least you know what types of method that you are going to do for your final year project if you plan to do the antioxidants. And then please make sure that what is the target of antioxidant that you focus, activity mana. Itu saja. For S2, please focus on the mechanism. S2, I believe that you have to find out what is the mechanism. So for S3, you have to focus on the activity and interactions of the antioxidant, what happened inside actually. Okay, boleh? Itu cara kita bagilah the level. Okay? Tapi kalau yang S1 boleh buat molecule docking, kita galakkan. Sangat digalakkan. Saya lagi happy. Itu impact dia sangat bagus. Okay? Okay, habis untuk that one. So, any questions from the participants from the Zooms or in this face-to-face audience? If you have any questions, you may ask now before we rest around 5 minutes before we proceed for the second workshop, second talk, okay, second lecture related to the smart packaging. Any questions? Wajib,
hal yang ingin dikasihkan dari metode ataupun hasil penelitian.
Just share for your, just share you punya antioxidants apa yang you buat. Alright.
Bila Bapak Ibu yang berada di Zoom meeting yang memiliki hal yang ingin didiskusikan terkait metode ataupun result dari ataupun basic principle yang menjadi kegunaan, silakan didiskusikan ke Dr. Rovina.
Malu-malu. Oke, ada dari Rada.
Oh, oke, Salman, silakan.